Well, thank you for coming today. Our speaker today will be Dr. Paul Schliekelman. Did I pronounce it correctly?
Yes.
And he's actually a statistics professor at the University of Georgia. I don't know that much about his background because with all the hectic, trying to juggle everything, I forgot to ask about it. But I'm really glad his talk is actually one of the talks that really exemplifies the popular stem talk theory. Because he's a statistics professor, but the talk will also be about genetics and biology, so it's interdisciplinary. And I'm hoping that students who are interested in both areas will be in attendance today. So without further ado, let's give Dr. Schliekelman a warm welcome, and we can get started.
Thank you. So I'm a stat professor, but everything I do is in genetics and evolutionary biology, everything related to biology, and this talk doesn't have much math in it, but it's all, like everything I do is mathematical. It's all math and computers kind of applied to biology, but biology has become very, much more quantitative and mathy than it once was. It used to be, if you were a biologist, you spent all your time in the lab doing lab work, and they still do a lot of that, but there's a lot more people who are biologists who are spending their time sitting in a computer analyzing data rather than generating the data. So it's, like I said, there's not much actual math in this talk, but it's all math underlying it. So I want to start just by saying, okay, so we're interested in talking about the human DNA sequence and how the sequence relates to different characteristics of us, like our height or weight, IQ, whatever. But I want to start by saying, what do I mean by a genome? And so when I say a genome, I mean, if I talk about the genome of an organism, then I just mean its full DNA sequence. And so those of you who've had some basic biology, there's the four nucleotides A, G, C, and T. So the DNA sequence just means that sequence of those four nucleotides. And the genome, you can think of it as containing the instruction manual for making the organism. So the human genome, our genome, it's around 3.3 billion nucleotides long, or 3.3 billion letters long. Among those 3.3 billion letters, there's something in the order of 20 to 30,000 genes. And it's the genes that actually do everything. Genes code for proteins. That's what a gene does, is it codes for a protein. And then proteins do most of the work that happens in a cell. And basically, everything that happens with the structure, function, like everything that we do in R is basically, I don't want to say R, but all the physical attributes are all resulting from the genes, more or less. And everything that's happening kind of on a day-to-day basis as you're going about your life, it's all happening because proteins are, genes are expressing proteins, and proteins are doing all these different things that need to get done in our bodies. Okay, so this shows a rough idea of the human genome and what it consists of. And if you look, this little sliver right here, which is only about 1.5% of the genome, that's the part that's mostly the functional part. That's actually like what we call the coding part. It's the part that actually codes for the proteins that do everything. Speaking loosely, most of the rest of it isn't functionally important. Like there's this huge chunk, the light green part that's, you probably can't see it, but there's the big light green part that says repetitive DNA. And so that's about 44% of the genome is this repetitive DNA that comes from something called transposable elements. And transposable elements you can sort of roughly think of as being parasitic genes. They're basically genes that all they do is copy themselves. So these things will get into a new, like for example, the last one, big one in humans was back before we split from chimpanzees, but around that time we got this transposable element got into our genome. It started copying itself and made enormous numbers of copies of itself. Kind of eventually what happens then is the genome evolves some way to shut that down because it doesn't want to be completely overwhelmed with these transposable elements. But anyway, this huge chunk of our genome is just these transposable elements, which again, essentially, they don't do us any good at all. And in fact, they probably hurt us in a lot of ways. But they're there just because they get in, they can copy themselves, and they can just make enormous numbers of copies. There is this other darker green, other types of repetitive DNA, which when I say repetitive DNA, I just mean the same bits of sequence repeated over and over and over again. So there's that 44%, which is the transposable elements. There's another, what is it, around 15% which is other types of repetitive DNA, and then and I should say these numbers are a little bit fluctuating. Like this particular graph is a little bit old actually. Some of these numbers have changed a little bit, but not a whole lot. But one of the interesting things about it again is just that how little of our genome is actually functional in the sense of being there to code for some particular thing that needs to be done, and like huge amounts of it apparently don't do anything at all. Although there's been some stuff recently which makes that question, that whole issue a little bit more complicated, but I won't get into that. But that's the basic idea of what our genome is like. This is a map, so to speak, of chromosome 1. So chromosome 1 is, so we've got 23 pairs of chromosomes. You've got for each chromosome you have the one you got from your mother, the one you got from your father, so and then 23 pairs total. So it's about 246 million base pairs long. So when I say base pair, I mean those letters A, G, C, T. And if you look, and again, you probably can't see it that well from where you are, but you've got all these things shown along here. And each one of those is a disease or some kind of condition. And what it's showing is all these different diseases or conditions where we know the locations of genes. And so basically genes are just, laid out along the chromosome there. And that's, again, showing for chromosome 1, again, just all sorts of locations where it's known that there's a gene at that location which affects, a particular disease. So again, you guys can't see it from way back there, but there's, colon cancer, neuroblastoma, glycogen storage disease, and all these, I don't know what most of those are, but they're diseases that people have done these. I'm going to talk about how they do this. They do gene mapping studies and found genes for them. Okay, a couple definitions. So the first definition is a genetic locus, and that's a specific location of a gene or DNA sequence on a chromosome. The plural of locus is loci. So again, that just means location on a chromosome. And then the variant of the DNA sequence at a given locus is called an allele. So you have different alleles at a locus. Like I said, the different alleles are different versions of the DNA sequence that someone might have at that locus. So when we go back and look at this previous plot here, and I have each one of these locations, each one of those would be a locus. So each one of those is showing a locus for a particular disease or condition. Okay, well, how does DNA vary among people? That's what I'm really interested in talking about today, is how does DNA actually vary among people? And this is showing on the left, well, it's DNA sequence, showing that, the double helix of DNA. And if you look, you'll see that we have this is showing two different individuals here. And the two different individuals are identical, like this one has CGAT, this one has CGAT, and so on, except there's one position here, which is marked as SNP. And that one position right there is different between these two individuals. And so that is known as a single nucleotide polymorphism. And that I'm going to call the SNP refers to a nucleotide position at which different people in the population are commonly different. And our definition of common is going to be 1%. So we call the SNP if we see that at least 1% of the population has something different than everyone else's. And that can vary anywhere up to 50%. So we're just talking about nucleotide positions where you will commonly see, so if I could choose any two people in the room, like I take me and you, for example, then at the SNP positions, there's a pretty good chance that we would be different at those nucleotides. And most of the positions, it's not that way. So now about 1 in 200 nucleotide sites in humans are SNPs, which comes out to be around 15 million. So it's a lot. Since there's around 3 billion nucleotides total, so then 1 in 200 turns out to be on the order of 15 million. Most of these SNPs don't do anything in the sense that it doesn't make any difference what you have at that SNP. Whether you have an A, G, C, T, it doesn't make any difference at all. And that's because most of the DNA doesn't do anything. So most of our DNA, like I showing on that one plot, most of our DNA doesn't do anything. It doesn't have any functional impact on us. And so for that reason, most of those SNPs also don't do anything. But some do. Some of those SNPs do. So some of those, if I have an A and you have a G, it can go and make some difference, like make me taller or make me have a higher probability of getting a disease or whatever. And these SNPs originate, the way the SNPs originate is as mutations. So what happens is different things can cause mutation, but different things happen. Say my parents have got, my dad has got his DNA sequence, that DNA sequence is passed on to me, but some kind of mistake happens, and so some nucleotides get changed. And so that's a mutation. Nucleotide gets changed. A lot of those nucleotides, a lot of those changes will just disappear. Like if I'll just have a change, and then, you know, my wife doesn't have that change, and our kids will each have 1/2 chance of getting that mutation. And in fact, you know, most of them just disappear, but occasionally they will just either by chance or if they have some kind of benefit, that they will become common in the population, and then that's when they become a SNP, is that they become common. But most of the time when a mutation like that occurs, it'll just disappear. Now, those are the common variants, and then there's also the rare variants. And so I talked about this, you know, 1 in 200 nucleotide positions roughly is a common variant where if you take any 2 people in the room here, there would be a pretty good chance that they would be different on those. But then there's the rare variants, which you might share with your family members, but you're not that likely to share with anybody else. And we don't actually have a good understanding of this yet. This is actually a really hot topic kind of in genetics, is this whole issue of the rare variation for reasons that I will get into at the end. So we don't have a good idea about the extent of the rare variation, but some very recent studies suggest that I said, you know, maybe 1 in 200 nucleotide positions are common variants, and then maybe several times that, 3, 4, whatever, 5 in 200 are the rare variants. And so again, if you go and look at me, for example, you would find that, okay, I would have some of the common variants that I would share with all of you, and then I would also have some rare variants. Only I would have, and nobody else would have. But then each person in the room would have their own selection of rare variants that probably nobody else shares. And then, so I've talked about the single nucleotide variants. There's also other kinds of ways that your DNA can be different. You can have deletions. And so this is showing a case here where here we've got A, B, C, D, E, F, G. And then in this individual, somehow that C and D got lost. And so that's called a deletion. There's a translocation where the DNA sort of gets scrambled between, so you've got a situation like this is the mother's DNA and this is the father's, and then it kind of gets scrambled around so that you end up with 2 chromosomes, the one that is actually missing part of the DNA and the other one that has gained DNA from the other chromosome. You've got inversions where here we got A, B, C, D, E, F, G, but then the C, D, E gets flipped around so it becomes E, B, C. And then there's also something called copy number variants where you've got bits of pieces of the DNA get copied, again, for various reasons. Various kinds of mistakes happen, and they get copied so that you get, again, you look from person to person, you will see how, like here, there's a duplication. So this bit of DNA right here, somehow by some mistake got duplicated, and so maybe in the father it was like this, and then the child it was like this, and then for reasons I won't get into, once that happens, once you start getting these multiple copies of things, it actually makes it easier for mistakes to happen again, because when there's multiple copies of things, and it tends to kind of confuse the kind of machinery by how it does replication and all that. And so sometimes these particular locations, you'll have a lot of variation, and you know, some people will have 20 copies, and someone else will have 22 copies, and someone else will have 18 copies, and so on. And about 12% of our DNA is the result of copy number, of this kind of variation, copy number variation. They can range from 1,000 nucleotides up to several million nucleotides in length. And like with the SNPs, most of these don't really have any effect in terms of, you know, our characteristics, but some do. And this has been another fairly hot topic in genetics in the last, you know, few years, about looking at these so-called copy number variants and how they impact our traits. But okay, so that's just a rough overview of how DNA, what our DNA is like, and how it varies between different people in the population. What I want to talk about today, the main focus today is what is different between people that makes them different in traits that we might be interested in, like height, weight, intelligence, disease occurrence, and so on. And I want to define the words genotype and phenotype. So phenotype is a trait or characteristic of an organism that we're interested in, like height, weight, IQ would all be phenotypes we might think of in humans. And of course, just about anything at all that you would care to measure could be a phenotype. The genotype is the genetic makeup of an organism or group of organisms with respect to some trait. So I'm going to talk about height as what I mean is all of the particular DNA positions that affect height. Okay, so I've talked some about how DNA sequence varies, and couple of points I want to make here. So the first point is that if you talk about why are species different from each other? Species are different from each other because they're different in their DNA sequences. All differences between species are due to differences in their DNA sequences. And so, no matter what you do to a dog is still going to be a dog. I mean, you can go and you can raise a dog exactly like a person, but they're still going to be a dog. So that it's completely the DNA that makes a dog be a dog. But now you talk about within a species, you talk about, okay, what's different? Like what makes all of us be different from each other? And that's a combination of both differences in DNA between us and differences in our environment. And so that's the next question then is how do things break out between genotype and environment? And I think this is maybe the only equation that I have in this talk here, but a very simple equation. So the P represents the phenotype, the G represents the genotype, the E represents the environment, and then the G, X, E represent, well, genotype times environment. And that means things where there's an interaction between the genotype and the environment. And what I mean by that is, so we talked about the G part of the effect, and that's the part of the effect which varies by genotype, but that effect doesn't depend on the environment. But an example of a G by E effect would be, so there's a known particular mutation which, if you have this mutation or have this allele, it will increase your chance of getting clinical depression as an adult, but only if you had some kind of major trauma as a child. So again, if you have some kind of major trauma as a child, then if you've got the wrong version of this allele, it will increase your chance of clinical depression as an adult, but it has no effect. If you just go through your childhood and you don't have any big traumas, like like a parent dying or something like that, then it doesn't have any effect. And so that's an example of a genotype by environment effect, where the genotype only has an effect if a particular environmental thing has happened. Okay, so let's talk about a specific example, human height. So I want to think about the genetic part of human height, and I want to think about the environmental part. And so when I talk about the genetic part, then what I mean by that is all the locations in the genome that have DNA sequence variation that affects height. And so all those locations, we don't actually know much about this, unfortunately, and I'll talk again quite a bit about height. But the number of locations, it could be hundreds, or it could be thousands, it could even be hundreds of thousands. It's something that, like I said, we don't know too much about, although we wish we did. But that's what I mean when I say genetic part. I just mean all the specific locations in your DNA where having a different nucleotide or different DNA at that position makes an impact on how tall you're going to be. And then there's the environmental component. And then that's just everything other than DNA that's going to affect your height. So that's going to be diet, exercise, chemical exposures, what your mother did, like while you were in the womb, whether she smoked and all that kind of stuff. So just everything other than DNA, that's the environmental part, everything other than DNA that could affect your height. So this brings us to this big nature versus nurture question, which I'm sure many of you have heard about this. And the big question is how important is nature, how important is genetics versus the environment? Nature is genetics, nurture is the environment. How important is genetics, how important versus the environment in determining different traits? And that question has become known as the nature versus nurture debate. And we can actually quantify this pretty well by using some math and some statistics along with data on the correlation between relatives. Yes. So my example here is height. So I've gone out and I've measured the heights of 30 adult men. So I've got measurements in inches there. And if I look at them, I first have something called the sample mean, which simply is I take up all the heights, I add them all together, I divide by the number of measurements, which I had 30 measurements, and that comes out to be 69.6, which is just under 5 feet 10 inches. So that's giving us the average height in the sample. So that's telling us one thing. So that's telling us about the average height in the sample. The other thing, but we'd also like to know more. I mean, just knowing the average isn't that much information. So we might also like to know something about how variable that is. So is everyone very close to the average? Is everyone very spread out from the average? Or exactly how are they distributed? And for that, we calculate something called the sample variance. And so if I've got here, what I have here is I've gone and I've already calculated the mean. It's my mean is the 69.6. And then for each individual, I go and I say, okay, this individual, their height is 68 inches. The mean is 69.6. So I subtract their height from the mean, gives me negative 1.6. this person was 64 inches minus the mean is 69.6, so their difference is negative 5.6. I calculate all the differences, and then I square them. And the reason why they square them is because you can show that mathematically that if you add up all these differences that they'll always sum to 0. So we square them to make them all positive. So since they're all positive, they're now going to add up to being something positive. And so we add those differences all together. We get 31.36. Actually, I'm sorry, we add these all together, and we get 353.4. And then we go and we divide that by the number of values minus 1. And forget about the minus 1. The minus 1 is there for esoteric reasons that I'm not going to explain. But the basic idea is that you're going and you are, for each individual, you're taking their difference from the mean, you're squaring it, and then you add it all up. And so it's and then you're dividing by the number of individuals, and that's giving you kind of an idea of what is the average square deviation from the mean. And so that's the variance. That's what's called the sample variance. And again, this is giving us a measurement of how spread out people are, how different people are in the sample are from each other. And so if everyone was exactly the same, if everyone was just exactly 69.6 inches, then the variance would be 0. If, on the other hand, if we had half of the men were 4'10", the other half were all 6'10", the mean would still be the same in that case. The mean would still be 5'10", but the variance would increase by up to 103. So in this case here, it was 12.2. That would increase it all the way up to 103. So the variance, like I said, just giving us a measure of the amount of variability, how different people are from each other in the sample. Now, we're back to this question of the nature versus nurture. And so you can do some math, and you can show that if you go and you say, okay, the phenotype is equal to the genotype effect plus environmental effect, then it turns out that the variance of the phenotype you can break in, it just simply turns out to be the variance of the genotypic part plus the variance of the environmental part. And then based on that, we define something called the heritability. And the heritability is, in the numerator here, this is that variance in trait due to genotype. And then the denominator here, it's the sum of the genotypic variance plus the environmental variance. And so overall, that's giving you a measure of how much so the denominator is the genotype variance plus the environmental variance. So that's the total variance. In the top, you have the numerator, you have the genotypic variance, and so it's telling you what portion of the total variance in the trait is due to genetics. So that's known as the heritability. So again, what that's doing is it's measuring, if you're asking what is different between people. So like, what makes me be tall? Like, what is different about me that makes me be tall? And specifically, how much of it is due to my genes? How much of it is due to, what I ate as a kid or whatever? This heritability is going to give you a measure of that, and we'll get back to exactly what that number turns out to be, what it turns out to be for humans. But that's what it's measuring is how much of what, like, for example, makes me be tall, how much of it is due to genetics. Okay, so the way we're going to get at this is by looking at the correlation between relatives. And so if you think about it, you realize that the stronger the genetic part of a trait is, then the higher the correlation in the trait between relatives should be. So again, if genetics doesn't matter at all, then, for example, there should be no similarity between me and my brother or me and my siblings. If genetics matters a lot, then we should be very similar in a trait. So again, you can go and you can use, yes. No, go ahead. Mm-hmm. Well, they're not necessarily equal. I mean. Well, I guess there's the G and there's the E, but the G could be bigger than E or smaller than the E or whatever.
Now show preference for genetics.
Well, so first, when I have the equation P equals G plus E, so that doesn't require that the G and E are the same size. So they'll just try to be what they are. I guess they do get equal weight, but they don't have to be the same size. G could be bigger than E, for example. And then when you get to the here, so here, it could just as well define it as heritability equals or it would be something other than heritability, but you could just as well define it, have something else that you would call would be the the environmental variance divided by the sum, and then that would be the proportion of the effect which comes from the environment. But,
Why did you do that?
I mean, the other one would just be 1 minus. I mean, that would just be 1 minus what I'm going to calculate here. So if I do this one, if you want to know the other one, you just take 1 minus this one, and it'll be the same thing. Okay, so like I said, my observation here is that the stronger the correlation is, or the more important genetics is, and the more correlation you should expect to see between relatives. And so this is showing here. I've got my top plot is a case where I've got high correlation. So you can think about that as the x-axis could be, we could be talking about pairs of brothers. So each point would be giving you the heights of a pair of brothers. And so, for example, here the first brother has a value of 15, the second brother has a value of 12, or whatever that turns out to be. So this is a case where almost in all cases, if the first brother has a low value, the second brother has a low value. If the first brother has a high value, the second one has a high value. This is where there's low correlation. And so here, you see a low value, and he could just as well have a brother with a high value, and so there's no relationship between the two brothers. So then you go and you do some more math, and you can show that if the row, which row is, I'll just call it P, the PXY, it's the correlation between relatives of type X and type Y, then you can make some certain assumptions, and you can do some math, and it turns out that the heritability, if you go and you calculate the correlation, for example, between siblings, then you take 2 times that, and that gives you the heritability. So if the correlation turned out to be 0.3, the heritability would just be 2 times that, would be 0.6. And so for both parent-child or siblings, it's just 2 times the correlation. For identical twins, the correlation between them will equal the heritability. Now, if we go back to this height example, and again, I've got my x-axis here is the first brother, my y-axis is the second brother. I've got their heights, and so again, each point here is giving, again, that this brother had a height of 73 inches, this one had a height of 68 inches, and so on. So that could be a typical data set, and then I would go and, again, there's some math you do. You calculate what's called a correlation coefficient, which measures, if it's 1, that means that they're perfectly correlated. If it's 0, that means there's no correlation at all, so there's no relationship between them. And for this one, the correlation, if you go and you calculate it for that data that I just showed you, it comes out to 0.37. Then if you take that times 2, that gives you then an estimate for the heritability, which in this case comes out to about 74%. So that would tell us then we're going to estimate that 74% of the variance in human height is due to genetics. So this is an estimate. It's not going to be exactly right, but it should be in the ballpark. And again, you can go do some more math or statistics, and you can get an idea of how close it is likely to be based on the sample size and various other things. But it's an estimate. So it should probably be pretty close to the right value, but it won't be exact. Now this shows estimates of the heritability for different traits. And so again, what this is telling you is for height, for example. So again, we ask the question, how much of the variation in height is due to genetics and how much is due to environment? So again, if we ask the question, what is different between people that makes them be taller or shorter, and how much of that is due to genetics and how much is environment, and about 80% of it for human height is due to genetics. Likewise, IQ turns out to be 60 to 80%. Obesity, so we're asking there, what is different about some people that makes them be obese? About 70 to 80% of that is genetics. Schizophrenia is 80%. Survival to age 85 is only about 30%. Survival to age 100, no one's actually measured this, but it appears to be a lot higher, which is kind of interesting. The genetics doesn't have a huge role in whether you live to 85, but it does play a much bigger role in whether you live to be 100. And then down here, I have some different personality traits, which you see the personality traits, and probably not surprisingly, are much lower heritability. And I don't, I'm not sure exactly what the definition of neuroticism is or extroversion. I mean, I can guess colloquially what they mean, but I just pulled these out of some paper, and I'm not a psychologist. But anyway, you see how those are much lower, like the achievement striving. Again, you can guess as well as me what that means exactly, but about 18%. And don't worry about the narrow sense versus broad sense. Just look at the broad sense is what we're interested in here. So this, to some degree, quantifies that question about nature versus nurture. That is, we have exact estimates here. So we have these estimates here saying that 80% of height is genetic, or 60 to 80% of IQ is genetic. Now, there are some caveats here. And one of the caveats is if you remember back when I said we can do some math and get this heritability estimate, I said under some fairly reasonable assumptions. But the fairly reasonable is somewhat in debate. And I'm not going to get into all the details of that, but just suffice it to say that not everyone agrees with those estimates for reasons that have to do with correlations between relatives that happen due to things other than genetics. And like I said, I won't get into all that. But I tend to think those are pretty accurate. I mean, for one reason, that there's a new method has come out for estimating heritability, which is based on advances in technology, which doesn't depend on those same problems, and those have tended to come out with about the same answer. So no one's done IQ, for example, yet, but like, for example, height by this new method turns out to be almost exactly what people estimated by the old way. And so from my point of view, probably those numbers in that table are probably about right. But I'll just say that not everybody agrees with that. Okay, so fine. So we've established now that genetics plays a very big role in a lot of different traits. But the next question is what? So we said genetics matters, but now what are the actual specific DNA differences that cause people to be different? So now I've said, okay, height is 80% genetic. So I can, based on that, I would say that, okay, I'm tall primarily because of my genes. That makes sense because all my family members are tall. My brothers, my sisters, my dad, my mom, everyone's tall in my family. So I mean, it's not a hard jump to say, okay, genetics is important there. But again, the question is what actual specific differences in my DNA make me be tall or make any of us have any particular characteristics? And the reason why I would like to know this is, for example, there's a lot of diseases that have a big genetic component. If we could understand exactly what makes people get the diseases, like what makes them be higher risk, we might be able to better cure them. We might be able to better know who's going to get a disease. This stuff also plays a big role in evolutionary biology and lots of other things. My biggest interest in it is and this is kind of gene mapping as a researcher. This is the stuff that I'm interested in. Yes.
So is this saying that
yeah, so for example, if we go back to that table, so schizophrenia is an example where again, if you ask what makes some people get schizophrenia and others not, about 80% of what makes you get schizophrenia, or what makes some people get schizophrenia is due to genetics. And so for example, if you're I think it's for schizophrenia, about 1%, in the United States, about 1% of the population will just if you just randomly choose people, about 1% of people will get schizophrenia at some point in their life. But if your mother or brother, if either a parent or a sibling has schizophrenia, I think it increases to 10. I think your risk goes up by a factor of 10 if your family members do. And that isn't necessarily all due to genetics, but because it could be other, shared environment. But a lot of that is definitely due to genetics.
When you had that up, so you said that it's 80% genetics, but you said you put that the heritability, so is environment factored into that number also?
Right. So the other 20% is the environment. So 80% is genetics, 20% is environment.
Gotcha.
Other questions? Yes.
I read this book called Genotype Diet. I don't know if you heard of it.
Say it again. Genotype what?
Genotype Diet.
No, don't know that one now.
Dr. Peter Giordano, he's a naturopath.
Yeah.
He said there's 6 genotypes. I mean, do you agree with that?
No. Roughly think about it, there's well, if we just kept the common variation, then it could be that there's, I know nothing about diet in particular. It could be that there's 6 common genotypes that affect diet. If you talk about all the possible genotypes, there's I would say it's almost impossible for anybody to make that claim because we actually understand so little about most of these things, which I'm going to talk about. Is there's almost no trait like diet that we even understand a tiny bit of the genetics. So I don't know anything particular about that, but just kind of in general, it's very hard to make that claim because there's almost nothing that we understand the genetics that well. But what time is this over? Okay. Oh, good. Okay. Okay, so the question is, how do we do gene mapping? And so this is showing, I've got, 9 individuals here. And let's suppose that the green ones have schizophrenia, the black ones don't. And then we see here that the red underlined nucleotides, those are the SNPs. So those are the ones where people vary. So the black ones are where nucleotide positions, everything except for the red underlined positions, you see that everyone's identical, but the red underlined positions are nucleotide positions where people are different from each other. Now, normally you wouldn't see this many this close together. I said about an average of about 1 in 200, so there wouldn't probably be this many this close together. But our question is then, is we notice here that the people who have schizophrenia, all the ones who have schizophrenia have an A nucleotide here at this particular position. Everyone who doesn't have schizophrenia has a G there. And that might tell us then that perhaps having that A nucleotide is what makes them have schizophrenia. Now, you could never conclude that based on only 9 individuals. You'd have to have vastly more individuals than vastly more people in your sample list. But that's the basic idea, is if we go and we saw that, okay, like I say, like everybody who has schizophrenia or people who have schizophrenia have a high tendency to have, for example, an A nucleotide. People who don't have schizophrenia usually don't have that A nucleotide. That would be giving us evidence that it is that particular change in nucleotide that's causing people to get schizophrenia. Okay, what we're going to do then, if we wanted to go and do this disease mapping or gene mapping, is we're going to go and we're going to get a bunch of SNPs spread throughout the genome, and typically you might have 500,000 SNPs when you're doing this. And you're going to get a sample of people, some with the disease and some without, and often this might be on the order of 1,000 people, say, with and 1,000 people without, something like that. You're going to find out which letter they have at each one of those SNPs. And so going back to the previous plot here, we've got, some people at this one, for example, have a G, some have an A. At this one, some have a T, some have an A. So every one of those SNPs that you're measuring in your experiment, you're going to figure out what does each person have. And then you're going to go and you're going to find out, and you again do some more math or some statistics, and find out whether there is a significant relationship between the genotype of that SNP and the disease. When I say significant relationship, I mean that it's a strong enough thing that you could say, okay, this isn't just due to chance. Like if I go back here, if I just had 9 individuals in my sample, it could just very easily be that it's just pure chance that these 3 people with schizophrenia happen to have that A. So just having 9 people wouldn't be nearly enough. I need a whole bunch more, and then I want to be able to say, okay, again, when I say significant relationship, I mean that it's strong enough that I can say, okay, this is definitely not due to chance, that this is because that there really is a relationship between this SNP and the disease. And that's the basic idea. So if I go and I find a SNP and it says, okay, this SNP is strongly associated with the disease, then I know that it could actually be that actual SNP, either that actual SNP or somewhere nearby on the chromosome, there is a SNP or some kind of DNA sequence variant that is causing these people to get the disease or be at a higher risk for the disease. Okay, this brings us to an idea of simple traits versus complex traits. And so a simple trait means traits that are caused by alleles at a single locus. So that means a single DNA position. And Huntington's disease, cystic fibrosis, albinism, and many, many other examples of diseases that are caused by a single locus. And then in contrast to that, there's complex traits. And those are traits who are caused by multiple, and multiple could be tens, it could be hundreds, it could be thousands or hundreds of thousands, but multiple different genetic loci, which again just means DNA sequence positions. And most important traits, so by important I mean the things that we all care a lot about, like height and IQ, and also most diseases that affect a lot of people, like psychiatric disorders, cancers, and so on. Most of those are complex. So the simple traits are usually rare. Almost all the simple diseases are rare, and so they're important to the people who have them, but they're not that important in a public health sense because not that many people get them. And so most of the simple traits are like that. Just a small number of people get them. But all the ones, the common ones that lots of people get, those are almost all complex traits. So again, caused by many genetic loci. So if we look back at the history of gene mapping in humans, people have been doing this since the 70s, or actually even late 60s. And we've been very successful at mapping the genes for those simple diseases and simple traits. So those ones that are caused by a single gene, we've been quite successful at those. And again, there's thousands of those. There's databases. You can go look thousands of those kinds of diseases where we know exactly, or maybe not exactly, but we have a very good idea of what the genetic cause is.
Oh,
yes.
Sorry, yeah, one of the online two types of sex genes. What about sickle cell anemia? Where does that fall?
That one is it's a single locus one, so just a single mutation. I think it's a little more complicated, but basically it is just a single gene that does it. Yeah, and so that's an example of one that we understand that one quite well what it is. Yeah. So like I said, so the single gene diseases we've been very successful at finding. But in contrast, there's been very little progress in the complex traits. And so I started in this field around the year 2000. And at that time, you could say pretty fairly that was almost total failure up to that point, in the sense that there had been around that time, maybe about on the order of 100 studies have been done of these complex traits in humans, and they found almost nothing, and in the few cases when they found something, they could never reproduce it again. And then some stuff changed, like at that time, due to technological limitations, the number of, well, it wasn't even SNPs. Like I said, when we do gene mapping studies now, we have these things, we have the SNPs, and we'll have 500,000 of them. We had something else that we used other than SNPs back then, and then you might only have at most a couple hundred of them. And then for certain technological reasons related to that, you had to do it in families where you would be looking at related people and comparing them. And it's really hard to get like large families of people to come and agree to be in a study. And so the studies were much smaller in terms of the number of people. They were much smaller in terms of the number of locations in the DNA you could look at. And then some stuff happened. There's the Human Genome Project you might remember, might have heard about, and some various other things that happened as a result of that. Suddenly we were able to have these SNPs where we could do 500,000 of them, or even 1 million if we wanted. And also because we don't have to deal in families anymore, we could just go out and just randomly sample people with the disease and without, and various other things that happened. Suddenly the scale of these studies got much, much bigger. And so in the 2000s, like between like starting about 2005 up to now, we've had these things called genome-wide association studies, where these studies where you go and you get a big sample of people with the disease, big sample of people without. You go and you genotype them on, say, 500,000 markers, 500,000 SNPs. And then you do like what I said, you go look at each SNP and you see, is this SNP associated with the disease? And at first, people were really excited because there have been these years of it never working. And then the first of these GWASs, which stands for the Genome-Wide Association Study, started to come out. And it looked like, and they were finding stuff. And it was like, awesome, this is great. This is working out like we expected because we're finally finding stuff. But then, well, this particular study I'm going to talk about was a big one. And there's a number of other studies that came out. This is kind of late, 2009, 2010, in that era, and where people started to realize it wasn't as good as they thought. And so this is an example. So this is a study of human height. So they had a sample of 13,600 adults of European ancestry. Each one they genotyped on 500,000, 500,000 SNP markers. They measured the height for everyone. They went and did the stuff that they do, and they found 20 of these SNPs that affect human height. So initially that seems great, like, all right, we're successful. We found 20 SNPs that affect height. But what these people did, which is different than a lot of other people did the studies, and they would just stop and they'd say, okay, we found 20 SNPs, let's publish our paper and say, great, we found some SNPs. But what these people did is they went further, and they took those 20 SNPs, and they took a new sample of 16,000 more people, and they genotyped those new sample of 16,000 more people with these 20 SNPs, and they asked the question, how well do these 20 SNPs predict height in this new sample of individuals? And it turns out to be not that well at all. This is showing, it's a figure from this paper, and so the x-axis here is the number of tall alleles. And so you've got 20 genetic loci. Each one has 2 alleles, one you got from the mother, the one from the father, so that's a total of 40. So you could have from 0 to 40 tall alleles. And you see that, and so the x, this is the number of tall alleles. This is showing, the gray is showing the number of people that had that tall allele. So for example, about 14% had 22 tall alleles. And then the lines, the blue dots, are showing the average height of people who had that many tall alleles. And so the bottom line, if you look at, like from here to here where most of the people are, there's hardly any difference in height. Like it goes from just a difference of about 2 centimeters. You look at again, you look at the effect on where most of the people are, and all those genes together only can predict about 2 centimeters of their height. So it only makes a 2 centimeter difference. And so if you figure it out, so it turned out then for these 20 genes or loci that they found, each of the alleles cause a change in height of about 0.2 to 0.6 centimeters per allele. So each tall allele you had would increase your height by between 0.2 and 0.6 centimeters, which is actually really disappointing because we've got this thing here. We've got a sample size of 13,000, enormous sample size. We've got this great technology of having 500,000 markers. There's some expensive project that was done that enabled them to get these markers and choose the right markers and all this. Everyone was really excited about this. It was supposed to be the thing that was going to, finally let us break open the complex traits, and all that together, and you can only account for 3% of the known genetic variation. Which, like I said, that's really disappointing because who cares about 3% really? I mean, what is 3%? You talk about a disease, and if you can explain 3% of the risk of the disease, that's useless in any kind of clinical sense. I mean, no one's going to develop a drug to change your risk of a disease by 3%. And so this paper is one of the big ones that really coalesced. Like people said, oh wow, this is not going as well as we thought. And then there was a number of other papers that came out around, other studies that came out around the same time, kind of come to the same conclusion. But that was the big one where it really coalesced. Everyone really realized, oh crap, this isn't going as well as we thought it was. So the question is why did it not work? And one of the possibilities is the sample size was too low. And so they had an enormous sample size of 13,000, but maybe it still wasn't big enough. And this next plot is showing, I think I'll skip this plot for time, and just go to this. So as I said, that study came out, and some other studies came out around the same time, all saying, look, we're getting these results, but actually they're adding up to being very little of what we know is there. And we know, again, like all these traits, we know the heritability by looking at, like what I talked about. We can go look at the correlations between relatives. We can estimate the heritability. So we know there's these big genetic components, but yet we do these studies, again, these enormous studies, and all we can explain is this tiny fraction. And so, and again, this has happened just again and again and again over almost all of these traits that all the complex traits that we've looked at. There's been, I think now, over 1,000 of these so-called GWASs for many, many different traits. And this clear pattern has come out that although they're finding stuff, almost all of them find stuff. They'll find, 10, 20, 30, whatever variants, DNA positions that are affecting the trait. They almost never add up to more than 10% of the known genetic component, and usually a lot less. And so the question of why this has failed, like why these GWASs have failed, has come to be known as the missing heritability problem. And so they call it that because there's the heritability, like we know, for example, that height has 80% heritability, but we're only able to explain about 3% of that 80%. The question is, where is all the rest of that heritability? Where is all that other genetic effect, and why can't we find it? And that's been, for kind of human gene mapping, that has been the big question. All the people like me who are working on this stuff, that's what everyone has been trying to figure out for the last 5 or so years. And there's a whole lot of possible reasons. Like one reason could be that you talk about height, for example, and height is determined by just a very large number of genes of very small effect. So maybe I shouldn't say genes, I should just say DNA sequence positions. So maybe there's like, who knows, 15,000 different DNA sequence positions that each one contributes just a tiny little bit to height. And if that was the case, if there's really that many and each one just contributes a tiny little bit, then we would just have to have like immensely larger sample sizes in order to be able to find the stuff we're looking for. So that's one possibility. Another possibility is that it's mostly determined by rare variants. And so for reasons I haven't told you, these gene mapping studies we've been doing, they're good at detecting the common stuff. Like I talked about the SNPs, where if you look at them and, a SNP, these positions where if you take any two of us, we would have a good chance of being different, those positions. That's the common stuff. Then I talk about there's also a lot of rare stuff, which is kind of unique to particular individuals. I mean, maybe not the only person in the world, but like I said, if I take the rare variants in me, I probably wouldn't share those with anybody else in the room. There'd be somebody somewhere in the world I share it with just by chance, but I probably wouldn't share it with anybody else. And so these gene mapping studies we've been doing aren't very good at getting those. So that's another possibility. And then there's all kinds of other things. It could be, for example, that I talked about at the beginning, that genotype environment where I talked about the gene where it causes you to have a higher chance of depression, but only if you had a major trauma as a child. Like if that sort of thing is really widespread, it would be very hard to find it because you'd never be able to find it except if you knew the right particular trigger, the right environmental trigger. And so that's another possibility. But that's kind of to some degree where we stand is that big disappointment because the GWASs have failed. And then the question is where next, and kind of where we are, where next is, well, part of it depends on what the cause is. And so we don't really know what the cause is. There's different people have different ideas about what the cause is. And you know, like I said, people like me and various other people are trying different ideas of how to go forward. But one of the big things that is happening right now, a huge revolution in biology, is whole genome sequencing. And so the first DNA sequence, which was published, so it was the first DNA sequence of a single human, and that was published in the year 2000, and that took about $3 billion, so billion with a B. That's a 1 followed by 9, or 3 followed by 9 zeros. So it took $3 billion, and it was hundreds of people in labs all over the world working for 15 years to get that single DNA sequence. Today, you can do the same thing. You can get a more or less complete DNA sequence. It can be done in a couple of days by a single lab technician for a cost of, it's getting close to $1,000. It's definitely less than $5,000. And so you've gone down from $3 billion down to maybe $1,000. And so, and everyone thinks this is going to drop even lower. It's just dropped tremendously, and people think it's going to drop under $1,000 in the near future. And so that just totally changes what we can do, because before we've been limited looking at these specific nucleotide positions. Now all of a sudden we're going to be able to look at the entire DNA sequence, and there's all kinds of reasons why that's important that I haven't told you, but a lot of it just comes down to, again, if I'm just looking at the particular nucleotide positions, that's all I can find. If my disease gene doesn't turn out to be close to one of those, then I'm not going to be able to find it. But if you have the entire DNA sequence, then whatever's there, you're going to have. But this also introduces all its own huge array of problems. And again, I won't get into all that. It's this summer there's the big human genetics meeting. It's like the big meeting for, the World Meeting for Human Geneticists. It was in San Francisco, and I was there, and everyone was talking about the whole genome sequencing, and people are really excited about it, and think, that this is going to be the solution to all our problems. And I'm skeptical for reasons, again, that I'm not going to get into. I mean, I think it's going to help us a lot, but a lot of it comes down to, like, if for example, it turns out that the reason why we can't find these things is because there's, the reason why we can't find the height genes is because, or the height variants is because there's 15,000 of them, and each one of them has a tiny, tiny effect. The sequencing isn't going to help. Yes.
You may know Gates, who is a historian, and he has taken a person's blood and been able to go back as African Americans to trace our ancestry back to early years
right.
before we were ever born.
Right.
So in that regard, could that possibly help people to know their entire genetic trace factors in terms of things that might be there that are complex that we would not otherwise know, especially as having come to America and not know where our heritage actually came from?
Yeah, well, I mean, you might, if you're not familiar with this, check out, there's the National Geographic has this thing called the Genographic Project, I think it is, where you can, I've done this, you take a cheek swab and you send them the cheek swab with I think it's $100 or $99, and they will take your DNA and they look at some markers, and they, from that, trace your ancestry. And so that's super cool, and it depends on where you're from and like where your ancestry is, exactly how accurate it is and how much they can tell you, but I know they've done a lot with all kinds of different populations. And then the more people who do it, so the more people who do it, then the better data they'll have, and the better they'll do it. And also, as the technology gets better and better and better, like right now, so it costs $99, and that's partly, they look at a fairly limited number of these sequence positions, but the cost of doing that is dropping so fast that it probably won't be too long before it'll be $99, and they'll be able to do much, much more detailed look at your DNA. But still, unfortunately, like right now, in terms of most of these complex traits, we just don't know. Like you can, I think this is on the next slide here. Well, anyway, you can right now send your DNA. There's that National Geographic thing, and there's also a company called 23andMe, and there's a couple of companies like that. Again, you send them your DNA, you pay them some money, and they will give you your tell you your disease risk and for certain known disease, and they'll tell you lots of different things about your genetics. I mean, it's to some degree maybe not worth the money, because again, it can for these certain rare diseases where it's just a single gene, like those are good, but the ones where it's like IQ or something like that, like we're so far from being able to explain predict IQ or know anything about IQ, because again, it's the same thing where we can only explain a tiny bit of it, but yes.
I have a question about how you calculate heritability. It comes from the variance in genetics, right? What are the two components of it?
The genetic and the environment, yeah.
So how do you calculate the genetic variance? Is that from everybody that's related?
I didn't really show you that at all. I mean, like the way that most of those numbers are actually done is that it's slightly more complicated than what I said, but most of those numbers I showed you, where they actually come from is they go and they measure what's called the correlation. They take identical twins and non-identical twins, and they measure the correlation like we're interested in IQ. They would measure the correlation in IQ between identical twins. They would measure it in the non-identical twins. And then you do some math, and it turns out that 2 times the difference of those 2 correlations is going to give you an estimate of the heritability. And so there's a lot of math in there. The reason why it's not very clear to you is because I didn't really explain it at all. And there's a whole bunch of math in that you get from this relationship between the correlations and how that gets to the heritability. Like I said, it's kind of, a lot of math is what it comes down to.
Is that like the fact that heritability is 80% in height, and we can only account for 3% of that, but if heritability is calculated by using only like people that are related to each other or correlation between relatives, then you could be just looking at the rare factors and not everything, so that the 2 numbers would correlate to each other if you're only looking at one specific gene pool. Does that make sense?
I mean, one caveat I should say is that those estimates only do apply to the particular population in which they're done. So that's an important caveat that I forgot to say. But in terms of the way that they're estimating it, I think is good. I mean, I think it's hard for me to explain the reason for it. Again, there's just a lot of math that goes into how you get to it. But basically, it's not affected by the fact that, I mean, because we're doing it only based on the correlation between the relatives. It doesn't play into the whole issue of the 3% and all that. So I think that the estimates of the heritability are good. Well, like I said, there's that really important caveat, which I forgot to say, was that it is only specific to a particular population that you do it in. So you could imagine, for example, if we're talking about IQ, that if for example, we lived in a society where at birth, every child was taken away from its parents, and they were all put into some situation where they all were just raised in exactly the same way, they got exactly the same education, everything was exactly the same, there would be no environmental variation, and the heritability would go up, because if everyone's getting just exactly the same environment, then there would be no environmental, it would be all genetics. On the other hand, if we did it and we took everyone away at birth, and this science fiction thing I'm constructing here, and half of them were just randomly, they flipped a coin for every kid, and half of them were made as laborers and got no education whatsoever. The other half were given this really awesome education and said, you're going to be like the thinkers of our society. In that case, the heritability will probably be a lot lower, because you'd introduce this massive environmental effect, which would just be the random chance of the coin flip. And so that kind of situation. So that heritability, it's not, I mean, it is, a lot of it's part of it is a function of biology, but it also depends on, it is not necessarily the same if you do it in different populations. But I went far afield from what you're I guess I got like 1 more minute here. I'll just say that. So the one interesting thing I'll leave you with is that with sequencing has gotten so cheap that people think that if you live in a country like the United States, that it would be entirely feasible for everybody to get their DNA sequence. Because if it gets under $1,000, I mean, $1,000 is nothing on the sort of healthcare cost. $1,000 isn't very much at all compared to like what healthcare things cost. And so people think that within 10, 20 years, maybe less, it could be that your doctor, like your DNA sequence is just going to be on file with your doctor, and you can have your, who knows, you might have a Google app with your DNA sequence or something like that, and it's just going to be there, and it's just going to be a very common part of everything we do. And we're sort of not there. I mean, in the sense, like what people hope is that you could have this, and you could go and you could be able to predict risk for different diseases, and you would know all the stuff about like maybe disease treatments, like you would tailor disease treatments according to what your DNA says. And this one's a more of a scary one, but you could potentially do would be, you could predict these different characteristics of a child at birth by just looking at their DNA sequence. That's the kind of stuff people talk about. The reality is we're so a long way from that because of, like I said, like for example, that we can explain so little, like height, we can only explain 3% of it. And so people worry a great deal about this, which they should, like this kind of stuff about, we think it's a good idea that we could predict IQ or something like that at birth, and I think it's probably not a good idea, but on the other hand, it could be good if you could know things about diseases and know, whether someone's going to get a disease or know that if they do get the disease, you should do treatment A and not treatment B. But all that kind of stuff, like I said, we're still a very long way from being able to do that. And then this is just the last thing here. So this is what I was talking about, that 23andMe. This is the website. You go there. I think it's on the order of $100 or something like that. You send them, I think it's a cheek swab probably, and they'll analyze your DNA, and they will give you this readout on giving you lots of different genetic information. But I'll stop there. Thank you.
And of course, are there more questions? I'm going to ask the online questions.
So typically taking that it's exceedingly frustrating.
I mean, no, it's been exciting. I mean, it's definitely been the case that people have been disappointed about the way this stuff has worked out recently. But at the same time, this whole thing with the whole genome sequencing, everyone's extremely excited about that. And so maybe 10 more years from now, it'll all have not panned out again, and who knows. But definitely, I mean, for me, it's fun on a day-to-day basis. I mean, I think, on the one hand, there has been a lot of frustration. On the other hand, we've also figured a lot of stuff out. So, yeah. Is there someone else over here? Yes. You mean with? I mean, like I said, you can certainly within 10 years, I would say, you could get your entire DNA sequence if you wanted. And right now, you can go to this company, and there's a couple others, and you can send them $100, and they will genotype you on not everything, but on particular genetic locations, and they can then tell you certain things about disease risk and so on. But like I said, the big problem right now is that we still don't know what we should be looking for. For like most of the traits we're really interested in, we don't know where to look to know. Like we can find your DNA sequence. Like we can definitely find your DNA sequence, but the problem is we don't know how the DNA sequence relates to the things we really care about for the most part. Again, for lots of these rare diseases, we can tell you that, like whether or not you're a carrier for some gene for a rare disease, but we cannot tell you very much about, are you going to get cancer or something like that. So the problem isn't being able to get the DNA. It's the problem is making the link between the DNA and the traits. Yes.
On the latter side, how many are you family?
In my family, 2 sisters, 1 brother. I'm the tallest. I have 3 kids. Yeah.
Are they also tall?
They're all young. So my oldest daughter is actually somewhat short, although she's caught up. But my son is my 2 younger kids are both tall. Yeah, they are.
Thank you. So you were saying the myths are where we can see that people most likely that's where they're going to be different. What about all the other positions? Is it a guarantee that all the other positions are exactly the same?
No. So that's the rare variation. So there's the SNPs, which is the common stuff where again, me and you have a fairly high probability of being different on the SNPs. But if we go elsewhere, and like again, in me, like I said, we don't know this very well yet. We're just getting the technology to do this right now, but probably, I said an average of 1 in 200 nucleotide positions are the SNPs, are the common variation, and then perhaps 1 in 50 positions or something like that are rare, where I might have a particular variation there, but you probably wouldn't. And like I might have, like maybe almost everybody in the room would have a G at one of these places. I would happen to have a G, or I mean, everyone would have an A and I would have a G, for example. But probably if we went out and sampled 1,000 people, no one else other than me would have that particular one. And then you would have something different. It'd be some other nucleotide position that you would be different than everyone else. And so if you looked at your family members, they would be pretty likely to have those same differences. So you'd share them with your family members quite likely, or many of them you would, but you wouldn't share them with, too many people in the population. If you looked at a big enough sample, you would find somebody else who had that same thing. But so probably any nucleotide position could potentially be different, and probably every nucleotide position is, I don't know for sure that's the correct statement, but probably every nucleotide position is different for somebody in the world, or some percentage in the world, but it's a fairly small number that are commonly different. Yes. Correct, yeah. I think for almost every disease, I could be wrong in the statement, but my impression is that almost every disease that I guess almost every disease that is of the simple variety that has risen to a high enough frequency that somebody's cared to go look. This probably isn't quite true, but let's just say that a lot of them are known. Very large numbers of them are known. Not all of them, but a very large number are known. I mean, there are just so many of these different rare things that only 5 people in the world at a given time have, or something like that. So it's definitely not the case that every single one is known, because there's so many different rare ones. But there's a huge number that are known. Oh, so you mean in a given year, how many new traits? I don't have a good guess at that. I know it's actually, so again, there's something called the American Journal of Human Genetics, which is the biggest journal for human genetics, this kind of stuff. And in the last couple of years, this has actually accelerated again because of the DNA sequences become cheap again. I mean, have become very cheap. And so there's actually been a lot of focus on that kind of stuff in just the last couple of years. So it's actually really accelerated. Like almost every issue has several articles that are, we have discovered the, genetic variant that causes such and such rare disease. But I couldn't give you, if I was just going to give a wild guess, I would say it's I don't even know if I want to give a wild guess. Let's say, yeah, I don't even want to give a wild guess, but less than 50, more than 1, I guess, somewhere in that range. But yeah.
Here's another question online. How does DNA play in sickle cell disease and the traits? She's actually a holder of the traits.
So I'm not a huge sickle cell expert, but I mean, I know it's a single locus, single gene position, and if you've got the 2 copies, you get sickle cell. So that means you got 1 from each parent. If you get a single copy, then you don't get sickle cell, and in fact gives you resistance to malaria, which is the whole interesting story of why, interesting from the scientific perspective of why sickle cell is so common, because, and it's believed that it's so common in people from African descent because they lived in places where malaria was really common, and malaria, killed a lot of people. And so despite the fact that if you get 2 copies, you get sickle cell, but it has this huge benefit. Depends that you get 1 copy, you're resistant to malaria. And so it is, I think, a pretty simple disease in that sense. It's just a single gene, 2 copies, you get sickle cell, 1 copy, you don't. I think there's probably I have a vague idea in my head that there's been a few other little details about it I think it's a little more complicated than what I just said, but I think that's most of it there. It's just that 1 gene.
The difference in amino acids just comes down to amino acids back to the carbon position being different from what it should be.
So is it a single amino acid? Okay, so the person here is saying it's a single amino acid difference. So basically the DNA then, each 3 nucleotides codes for an amino acid, and so one of those amino acids, I don't know do you know if it's just a single DNA change, or do you know what it is, or? So it's just a single nucleotide position, I guess, which makes that amino acid change, which then changes the protein and so on. Yes.
So does epigenetics represent the environmental factors that obfuscate the research?
Epigenetics, that's what you said. You said epigenetics. Yeah, that's I didn't say that. And so what epigenetics means is it's been another area that has been really hot in the sense that people didn't really understand it very well up until recently. But what epigenetics means is there are certain changes that can happen to your DNA which are not, which are not permanent, which can be environmentally induced. And so things can happen to you that changes your DNA, and that can actually be passed on to your children, which kind of goes against like what you learned in, at least when I was a kid, I learned biology. You said, there's no possibility of environmental changes, being passed. Environmental. Like you remember the Mendel versus I can't think of his name. The inheritance of acquired characteristics. What's the guy's name? Lamarck. Yes, Mendel versus Lamarck, and how we proved that the things that happen to you cannot be, like environmental changes, can't be passed on to your kids. But it turns out that's not true. That we've discovered relatively recently that there's these things called epigenetic changes which can be inherited. So something could happen to me. it changes my DNA in a way that does get passed on to my kids, but it's not permanent. So it might last for a certain number of generations. It could just disappear with me, or it could pass on to my kid, or it could go for 3, 4, 5 generations, but it's not permanent. And so that is also, like when I talked about the missing heritability, and I talked about the different things people talk about, that's definitely one of the things people talk about. Now, my former supervisor, I spent some time at UC Berkeley, and my former boss there had a real interesting study, which I won't get into details again, where they seem to conclude that it probably wasn't epigenetics for reasons that I can't explain here, but it's probably not epigenetics was what he concluded, but that definitely, epigenetics is definitely one possible cause. And that's one of those things that we don't have like a great way to deal with epigenetics, kind of in the gene mapping. I mean, to some degree, these epigenetic changes, to some degree, they're not that much different than anything else in the sense that it changes the DNA. And then in that sense, it's no different than any other nucleotide change or whatever, but they have the characteristics they don't stay forever. And so the fact that they don't stay forever means that they don't necessarily get inherited, and so it kind of can potentially throw out the gene mapping. It could potentially mean the gene mapping doesn't pick them up, but it might pick them up. But again, yeah, that's a whole complicated area to which we don't know the answer completely. But my best guess would be that it's probably not epigenetics, but I could be wrong.
Any other questions? Well, if not, I have one last question.
Okay.
I have questions you were mentioning, student height. How tall are you?
I think 6'5", 6'5"-ish, yeah.
Okay, well, thanks, and thank our speaker again.
Thank you.